Thank you for the very kind introduction. So today I'm going to be talking about my master's thesis work on the influence of experimental silviculture on plethodontid salamanders. Before I get started, I'd like to make several acknowledgements. First, I'd like to thank my committee members, Drs. Carla Haas, Drs. Nicholas Caruso, Mike Ost, and Holly Kinsvater for their support. And of course, I wouldn't have been able to do all of this work without the incredible people here who helped me along my journey. So thank you, everyone in this room, and thank you for coming to listen to my talk. My projects take place in the Central Appalachian hardwood forests of North America. These are mesic hardwood forests that are common along the Blue Ridge and Central Appalachian Mountains, and they provide a source of valuable timber for our local communities. In the terrestrial ecosystems of the United States, salamanders often comprise the majority of vertebrate biomass. And they can actually be 2.6 times that of bird biomass. Despite their abundance in the eastern US, from 1980 to 2009, less than 6% of wildlife research journal space was dedicated to studies of herpetofauna, with reptiles making up the majority of this small percentage. And this underrepresentation highlights the need to conduct more studies on salamanders. Although we need more research on salamanders in general, we do know how important they are in the forest ecosystem. They influence leaf litter decomposition through the consumption and alteration of invertebrate communities, which can then influence carbon storage. They are highly efficient at converting their invertebrate prey into biomass, providing a readily available source of high-quality protein for other vertebrate predators. So now that we understand their ecological role, these guys are in trouble. Amphibians are one of the most threatened taxa in the world, with 41% of species considered threatened, the highest among all other vertebrates. And this decline means losing what these species contribute to the ecosystem. My studies focus on plethodontid salamanders. They're a family of lungless salamanders which respire cutaneously, and this requires their skin to be moist for gas exchange. They can desiccate easily and can be found along mesic forests or along streams. And they hide under cover objects such as rocks or logs, which retain moisture when surface conditions become too hot and dry. The primary threat to amphibians is habitat modification, and timber harvest represents one type of this modification. It involves the removal of vegetation, which can increase surface soil temperatures, air temperature, reduce soil surface moisture, and leaf litter. And this can negatively impact plethodontid salamanders. It's well known that salamander abundance declines after timber harvest in the short term. These salamanders have very small home ranges, and they don't really move around very much. So remaining in harvested areas could impact their body condition and because of fewer foraging opportunities. But we can't just do away with timber harvest. It forms the basis of many local economies, and we rely on it for infrastructure and other wood products. Forests should be managed not only to generate revenue, but also to regenerate trees and balance multiple uses, which impact salamanders. Currently, several mitigation strategies exist to reduce the impact of harvest on salamander populations, such as retaining coarse woody debris, but sometimes the retention of coarse woody debris only increases salamander counts in dry areas, or it first must be highly decayed in order to then benefit the salamanders. Sometimes the use of alternative harvest practices can mitigate some of the negative effects, but it could be beneficial to develop new strategies to retain higher abundances of common salamanders immediately after harvest and allow populations to achieve their pre-harvest abundance levels more quickly. So these are the three chapters in my thesis that I'll be talking about. Chapter 1 is on the long-term effects of silviculture. Chapter 2 is on the impact of artificial tip-up mounds, and Chapter 3 compares two different body condition indices. Silviculture differs from timber harvest in that it's the science of cultivating trees to meet management objectives. So a management objective could be timber harvest, or it could be creating wildlife habitat, restoration, or others. And this is an example of a silviculture prescription, but of course, these will look very, very different depending on the tree species and the management goals. The Southern Appalachian Silviculture and Biodiversity, or SASAB project, is a project that started in 1994. SASAB is a longitudinal experiment, which allows for a controlled testing of different treatments and a direct observation of actual differences over time. This study is unique and incredibly valuable because most similar studies are chronosequences, which are a set of similar sites that represent different ages, or the other studies are often over a shorter term. The original goal of this project was oak regeneration and the impact on biodiversity. SASAB spans a total of 4 sites in Virginia. Blacksburg 1 and Blacksburg 2, or BB1 and BB2, are located in Montgomery County, Virginia, in the Eastern Divide Ranger District. Clinch 1 and Clinch 2, or CL1 and CL2, are located in Wise County, Virginia, in the Clinch Ranger District. At each site, 7 different silvicultural treatments were randomly assigned and applied from 1994 to 1998 across adjacent 2-hectare experimental units. And just as a side note for people who don't think in hectares, 2 hectares is 0.008 square miles. So first is the control, in which no treatment was applied. The mid-story herbicide treatment, where an herbicide solution was applied to the bottom of stems. Group selection, in which stems were removed from 2 to 3.5 hectare openings. The shelterwood, which was a partial harvest that removed 41% of the basal area, with a stand re-entry at just 2 sites, BB1 and BB2, from 2007 to 2008. The leaf tree, in which 25 to 45 high-quality dominant trees were retained per hectare. The commercial clear cut, in which all merchantable stems were removed, but low-quality stems were allowed to remain. And lastly, the silvicultural clear cut, in which all stems over 5 centimeters diameter at breast height were removed. The goal was to determine whether salamander relative abundance had recovered in each of the silvicultural treatments 30 years post-harvest. Other studies predicted it would take 60 to 100 years for plethodontid salamanders to recover after harvest. To answer this question, we established 9 sampling transects in each experimental unit, measuring 2 meters by 15 meters in a 3x3 grid arrangement. We randomly selected 1 transect per treatment type to be sampled each sampling night, and we crawled along these transects on our hands and knees, visually searching for salamanders on rainy nights when the conditions were suitable for salamander activity. We counted number of salamanders and made note of species at each transect. And there's me looking for salamanders. So to analyze these data, I created a series of 7 negative binomial generalized linear models to investigate the impacts of silvicultural treatment on the relative abundance of salamanders over 30 years. I calculated relative abundance by taking the total salamanders in each survey occasion divided by the number of salamanders, or number of surveys. I used AICC for model selection and used the top model to generate a predictive plot of relative salamander abundance across all decades for all sites combined. And lastly, I created 2 different pairwise post hoc comparisons to determine statistical differences in relative salamander abundance, both between decades in each treatment and differences within decade among treatments compared to the control. From 1994 to 2023, we captured a total of 12,036 plethodontid salamanders across 1,883 sampling nights. So this figure shows relative salamander abundance on the y-axis and silvicultural treatment on the x-axis. And this graph is just for Blacksburg 1, but the results were similar across sites. The data is broken down by decade, to compare pre-harvest to 10 years post-harvest, 20 years post-harvest, and 30 years post-harvest. So for all sites, the relative abundance of salamanders in each treatment 30 years post-harvest is similar to that of the relative abundance pre-harvest, except for the shelterwood. And this is only for the sites that had a second stand reentry. And these results are really interesting because until now, we haven't really known when salamander populations recover after harvest, at least for plethodontid salamanders in this region. And similarly, looking at the control compared to the treatments just 30 years post-harvest, we can see that the control is similar to each treatment, except for the shelterwood once again, for the sites with the second stand entry. And this is important to consider because, of course, the environmental conditions vary every year. So when we're looking at the control compared to the treatments in the same decade, we're looking at salamanders that were counted under the same weather conditions. So our top model included decade, treatment, site, the interaction of decade and treatment, and annual precipitation. And we can see that the predicted relative abundance of salamanders by survey 30 years post-harvest appears to be similar to the pre-harvest, except the shelterwood. And again, this is really cool to see because we can see that the salamanders really have recovered, at least except the shelterwood. And this wasn't yet previously known for plethodontid salamanders. We really mostly only had predictions. So for nearly all treatments and sites, relative salamander abundance has recovered to the control levels 30 years post-harvest, with the only exception being the shelterwood. And this is likely due to the second stand entry from 2007 to 2008. And this means that it's only been 15 years since the last canopy removal at those sites. This finding is notable because it means a substantial decline in salamander abundance is likely after each stand reentry. This suggests it will likely take much longer for salamanders to recover from silvicultural treatments that involve multiple stand entries than a silvicultural treatment such as a silvicultural clear cut, which only involves one stand entry. Overall, forest managers should carefully weigh whether or not to implement harvest prescriptions calling for multiple stand entries in areas with sensitive or threatened plethodontid salamander populations. Also, we don't yet know the mechanism of recovery. Salamanders from surrounding unharvested forests could have gradually immigrated to the harvested sites. They could have reemerged from underground to forage more often as habitat became more suitable, or the remaining salamanders eventually reproduced enough young to replenish the population, but we're not really sure. Future research should aim to investigate the impacts of silviculture in other geographic areas over the long term with a variety of different site conditions, since we know that this can impact how salamanders are affected. Overall, this is a really encouraging finding and suggests that it's possible to balance some forest management using most silvicultural methods in this study and long-term plethodontid salamander population viability, which is amazing. So in forest management, we aim to simulate natural disturbances to facilitate the regeneration of different tree species. Some types of natural disturbances, such as wind throw, are not well mimicked in the current silvicultural techniques. Young forests often have less coarse woody debris and less pit and mound microtopography. So we're missing these structural elements of older, more mature forests on our landscapes that are added from wind throw. So does adding complex microtopography from wind throw benefit salamanders? So tip-up mounds occur when a tree is pushed over by wind or an ice storm, pulling up the root ball and creating long-lasting pit and mound microtopography. And these are the different parts of the tip-up mound. First, we have the pit, the mound, and the tree bowl, or the stem of the tree. The formation of tip-up mounds is an important source of disturbance in eastern oak forests. And my previous research findings indicate that in a Michigan northern hardwood forest, artificial tip-up mounds appear to positively impact salamander count. And this is from an undergraduate research project that I did at Michigan Tech. Other than my previous research, to my knowledge, no other study has looked at how tip-up mounds impact salamanders. However, we don't know why tip-up mounds could benefit salamanders and how they affect the broader forest community. So natural tip-up mounds have a lower soil temperature and higher soil moisture in the pit. They can provide habitat for invertebrates, can increase plant diversity, and the creation of tip-up mounds can increase coarse woody debris. This study was designed to be a follow-up study on my Michigan tip-up mound research. The goal was to determine whether artificial tip-up mounds impact salamander abundance in a central Appalachian hardwood forest and the potential mechanism behind any changes. To answer this question, I established 6 replicates each of 2 different silvicultural treatments, the control and artificial tip-up mounds, in the Fishburne Research Forest in Montgomery County, Virginia. Each treatment unit was 0.05 hectares in size due to space constraints in the Fishburne Research Forest. We randomly assigned plots to be either control or tip-up, and within each treatment, I established a 3x3 coverboard array, with each board spaced 1.5 meters apart. The boards were made of 2.5 centimeter thick white oak boards that were cut to 0.3 meter by 0.3 meter lengths. I allowed the boards to weather in place for 1 month before pre-treatment sampling. I created artificial tip-up mounds by using a compact excavator crane arm to push over 8 trees in each tip-up mound treatment unit. And I implemented tip-up mounds during the winter to minimize disturbances to any salamanders that would otherwise be active on the surface, as well as to reduce soil compaction. And there's me and Tal from forestry using the excavator to make the tip-up mounds. So first we looked at microclimate. I hand collected air temperature, humidity, and soil temperature data at flat areas, pits, and mounds at each treatment unit. And this figure shows the hand-collected microclimate data from the month of August, with soil moisture on the top panel and soil temperature at the bottom panel. And at both tip-up mound and control treatments, there was no difference in temperature or moisture at the coverboard arrays, which are the flat areas, which was expected. In the summer, the mounds were significantly hotter and drier, and the pits were significantly moister and cooler. I also placed temperature loggers in at least 1 pit, mound, and flat area located 2 meters to the north of each pit, and every single tip-up mound treatment unit to collect soil temperature data that could explain any potential differences in salamander habitat utilization. And this figure shows that data with date from August to December on the x-axis and temperature on the y-axis. And this logger data gives us a more comprehensive timeline of how the temperatures at flat areas, pits, and mounds change over time and seasonally. And we can see the temperature at the mounds really fluctuated a lot more than the flats and the pits, with the mounds generally hotter than other locations in the summer and colder in the winter. From August to about mid-September, the pit was consistently cooler than the flat. So we can see that temperatures and moistures differ between pits, mounds, and flat areas. Does this impact how salamanders utilize habitat? To answer this, I conducted 5 nighttime area-constrained searches at each pit, mound, and a corresponding flat area and tree bowl on rainy nights from September to November of 2023 to detect any potential differences in microhabitat usage. During these events, I had 2 pairs of observers look at our areas of interest, which were the tip-up mound, including the exposed root ball, and here you can see a little slimy salamander utilizing the mound habitat. We also observed the pit, a flat area of the forest floor, and the bole of the tree, because the salamanders do climb up the trees. This figure shows the location of salamanders on the x-axis and salamander count on the y-axis. And it turns out that despite temperature and moisture differences between flats, pits, and mounds, none of the salamander counts between any location significantly differed. But did the implementation of tip-up mounds impact total salamanders found in treatments overall? I sampled plethodontid salamander abundance and diversity weekly by checking under the cover boards at each site before tip-up mound implementation for one field season, and I conducted post-treatment salamander sampling using the same weekly cover board surveys for 2 field seasons. I counted the number of salamanders for each survey and marked each individual salamander captured during cover board surveys with a unique visible implant elastomer mark with the goal of estimating the population abundance using mark recapture. I caught a total of 59 plethodontid salamanders over 26 cover board surveys from 2022 to 2023. Our captures consisted primarily of eastern redback salamanders, with the remaining captures being northern slimy and two-line salamanders. And this eastern redback salamander is sort of an unusual silver morph. Most eastern redback salamanders have like an orangish or reddish stripe, but this one is silver. And we found 3 or 4 of these silver morphs at our study sites, which is just a cool side note. This figure shows treatment on the x-axis and salamander count on the y-axis. And salamander count significantly decreased after tip-up mound creation in the tip-up mound treatments. So salamander count declined on plots with artificial tip-up mound treatments, but not on control plots, and this was very unexpected. So the question is, why did this happen? This counters my previous findings in Michigan, which shows that eastern redback salamanders had higher counts in artificial tip-up mound treatments. So there were some key differences between the 2 studies that could have contributed to the conflicting results. So firstly, the Michigan study sampled salamanders over a much longer period of time. Also, the climate in Virginia tends to stay warmer, and even though we did create artificial tip-up mounds in the winter to reduce compaction, the ground was probably not frozen. Also, there was a large difference in the number of tip-up mounds per hectare between this study and my Michigan study. In this study, I actually pulled down 160 trees per acre versus the Michigan study, which only pulled down 14.7. And this is mostly because of how compressed and small our plots had to be, even though it was only 8 trees per treatment unit and ended up being many trees per hectare, just because of how small the sites were. Salamander count did not differ between those found on pits, mounds, flat areas, and tree bowls. Compared to flat areas, pits tended to buffer environmental conditions, and mounds tended to exaggerate them. During the summer, mounds were consistently hotter and drier, and pits were consistently cooler and wetter than flat areas. As the seasons changed from summer to winter, at some treatment units, pits were warmer than flat areas or than mounds. And I think this is probably because they retain more moisture, and so they cooled more slowly. Our results suggest that further study on how tip-up mounds influence salamanders over the long term is needed. Other researchers should consider the removal of the tree bowls after tip-up mound creation, so not to confound the effects of tip-up mounds with the effects of large coarse woody debris. Future studies should examine this topic on a much larger scale across different geographic regions and for a longer period of time to discern the potential impacts of tip-up mounds on salamanders and other taxa. So canopy removal can impact salamander body condition by making service conditions less suitable and allow for fewer foraging opportunities. More food generally equates to more body fat and a more favorable body condition. And these lipid stores can be used to assess the body condition of salamanders. Currently, most scientists use the residuals of an ordinary least squares regression of mass on body length to estimate salamander body condition, or the scaled mass index. But some have also used the residuals of an ordinary least squares regression of tail width on body length, because salamanders use their tails to store fat. Notably, only one prior study has actually compared these 2 methods, and they found that the index using tail width was not a reliable proxy of body condition. There have already been several studies that have utilized tail width in their body condition indices, so this warrants more comparison. So are methods using SVL and tail width reliable measurements to estimate body condition? To answer this, I conducted measurements on 49 eastern redback salamanders collected and cared for by our collaborator, Charlie. He fed the salamanders at two different levels in the lab conditions for 12 months, so we did expect to see differences in their body conditions. The salamanders were housed individually using plastic containers with moist paper towels and stored in growth chambers. I measured all salamanders on the same day with the help of 8 total observers with varying levels of experience. For each animal, 4 observers were randomly assigned. 2 observers were randomly assigned to measure snout to vent length, and 2 observers were randomly assigned to measure tail width. Each observer measured snout to vent length and tail width twice, but without knowing whether it was the same animal that they were measuring. And I measured mass once. I compared the standard deviation of centered and scaled snout to vent length measurements to centered and scaled tail width measurements to determine which measurement had more variation. I also compared the correlation between the same and different observer measurements for snout to vent length and tail width. So this is the graph that shows the centered and scaled snout to vent length measurements compared to the tail width measurements with a line, a 1-to-1 line. So if each measurement was exactly equal, they would all follow this 1-to-1 line. But you can see that there are actually more measurements that are over the line above, like closer to tail width. So this means that there were more tail width standard deviations that were larger than the snout to vent length standard deviations. So observer measurement correlation. Snout to vent length measurements by a single observer were the most correlated, with an R value of 0.9. Snout to vent length measurements by different observers were actually still higher than tail width measurements by the same observers. And lastly, tail width measurements by different observers were not very correlated, with an R value of only 0.45. Overall, snout to vent length is a much more reliable measurement of body condition, since these measurements had less standard deviation compared to tail width. And also, even different observer snout to vent lengths were more correlated than the same observer measuring tail width. Far fewer studies have used tail width as an estimate of body condition rather than mass, and only one other study actually compared the two. More studies have used mass and snout to vent length, so it's preferable to utilize an index that can be more easily compared between studies. Also, there is far less variability associated with snout to vent length. Therefore, I recommend researchers use snout to vent length and mass rather than tail width when conducting or calculating plethodontid salamander body condition. The next step was applying these findings back to Chapter 1. I plotted the residuals of a linear model of log mass on log SVL by treatment and decade, and it looks like treatment and decade doesn't impact salamander body condition. But this is in line with previous findings from this study, which found that treatment impact body condition only for salamanders at our BB2 site and no other sites. Take-home messages. Silvicultural treatments may not be as detrimental to long-term plethodontid salamander populations as originally thought. Salamander count decreases in the short term after artificial tip-up mound implementation in central Appalachian hardwoods. And snout to vent length measurements are less variable, both between the same and different observers than tail width measurements. So I suggest that scientists use mass divided by snout to vent length as a body condition index for plethodontid salamanders. Questions?
I guess we didn't test the microphone, so folks on Zoom, I guess if they want to get it in the chat, but
Simba?
Thank you for the talk, Tori. Super cool. I was wondering what the generation time is for the cell makers. How many generations you think roughly would have passed in this 30-year period?
Yeah, so usually they're about 3 or 4 before they become reproductively mature. So sometimes it takes females a little bit longer. So they can start breeding when they're 3 or 4 and start producing babies. And then those babies take 3 or 4 years to then grow up. Um, so I guess it probably took about 10 years for the same, like for the population time. Does that kind of
Yeah.
answer your question? 10 generations, maybe. So they can live a lot longer in captivity. There have been like a lot of like anecdotal instances of like eastern redback salamanders or other plethodontids living to be like 32 years old or 36. But in the wild, they don't really live as long. I think they only live um, like 8 or 9 years. Yeah. Hmm?
So you were in fish farms. Was it the southern facing aspect or the northern facing aspect?
I think it was southern facing. The sites were very dry.
Yeah, I'm curious if the aspects of my how that impacts the similar kind of
Yeah, so some studies have found that if the site conditions are already like very, very good for salamanders, so maybe they're like a northern facing aspect and they're very moist and wet, that can kind of buffer some of the negative impacts of silvicultural treatments. Whereas if the site is already not super great for salamanders, maybe has southern facing aspect, it's really dry and hot, it kind of exacerbates those impacts. So that could have also played into it.
I'm wondering if there's another way to create that microtopography with, I mean, obviously it'd be a lot harder. To not use an excavator, but shovel.
We thought about it. We did consider that. But realistically, looking at time constraints and also money, we were like, okay, so how many technicians are we gonna have to hire and pay to dig holes with me? And looking at the cost of renting an excavator and the time that it would take, and we're like, well, the Michigan study, they use forestry equipment and they went into the stand. So we're gonna stick with that. So that was ultimately what like made the final decision for us. But yeah, it would definitely be interesting to see because I think soil compaction really played into the results. Connor?
Really nice work. I know that you didn't look directly at this in your study. I'm curious on the SASAB stuff if you had any like anecdotal thoughts or predictions on. It seems like relative salamander abundance bounced back after that 30-year period. Do you think that species diversity was doing the same thing, or was there some species? I know it was mostly redbacks you were finding, but were you finding at least the same number of species across time periods, or do you have any?
Just kind of anecdotally sifting through the mountains of data in our database. I didn't notice anything that looked like odd or different about what species that we were finding, and honestly, we've trained our technicians so well that they always come to me whenever they find interesting things. So I feel like one of them would have mentioned that to me, but I personally haven't seen any differences. But I did not conduct an analysis on this due to time constraints.
Cool.
Hmm?
I was thinking also about the CSAP study, and it looked like, at least in the prediction plot, the control plot abundances also went down in the 10 and 20-year decades, and then maybe not significantly. I was wondering if you just thought that was sampling variation, or maybe there could be an effect of being adjacent to a plot that's impacted by silviculture. though they want harvest specifically, maybe there's stuff.
Maybe we can go back. We. No,
Tori wasn't here when the initial.
Right.
Implemented, no, like Mike and I know, that some of the skid trails did run through the control plot, that it was not like preserved as pristine. So there definitely was some.
Nice that it bounced back though.
yeah, especially like if sites are close to a treatment that removed a lot of canopy, you definitely get edge effects that are coming into the control. Transects that are near those edges could be a lot. These conditions impact salamander abundances. So that's what I would guess.
If follow up to that, are these the 2 hectare plot?
Yeah.
Is that, how comparable is that to what standard is our for actual implementation in the field? Because it varies
They're a
lot.
little bit small. It can range. It kind of depends on where you are in the world. A lot of people locally do the commercial clear cutting, which is high grading. That's what it is. But like say out west, clear cuts are much larger. So it really just depends on like where you are in the world and who's doing the management and for what objectives.
Sorry. I'm curious if observe any differences in so I don't know if you didn't see any differences.
Let's go to those plots because I can just pull them up. Oops. 'Cause I have them at the bottom here. The different treatments. So here we have, yeah, I do need to share the screen again. Actually, thought this was going to be easier this way.
You can't see it on Zoom.
Yeah, I got to tab over. Oops. Oh,
Well, we can see this.
Rewrite your plots from memory, right?
all right. Well, I might just have to. Oh, and now. All right. Well, we might just have to. Oh. See, every time I find it, it goes back to the very beginning. All right. Well.
Yeah.
Okay, here we go. So these are the Wise County sites. And it seems like they're pretty comparable, maybe even slightly higher than these Blacksburg 1 and Blacksburg 2 sites. And I found the same thing just anecdotally when I've been in the field. These sites, they're super wet, really great microtopography, and there's just a lot of salamanders that come out there. So I feel like there could be slightly more salamanders at the Wise County.
But they are high elevation. sites, so
Yes.
they're not affected. There's no.
Yeah, they're pretty left well alone, which is nice.
You have a question in the chat from
Mhm.
Joe Buckwalter. He says the plots shown previously seem to show a strong effect of silviculture. Abundance at controls jumps from 9 to 13 over 3 decades. But this increase was not seen in the treatments. But the stats did not bear this out, I guess is the question.
Hold on,
The-
let me,
I think that was on. Their slides.
because I realized the Zoom people can't actually see.
Is it still not showing?
This isn't.
No, it's not shared. Okay. Okay, so maybe it'd be easier if I just pull up the chat. Okay. Shown previously seemed to show a strong effect of silviculture. Yeah. Abundance at controls jumped from 9 to 13 over 3 decades. This increase was not seen in the treatments. Stats did not bear this out. Okay, yeah, so I think I understand the question. Yeah, you can see that the box plots and the control overlap. So there were some versions of the model that we ran where it looked like the control levels were increasing, but it wasn't statistically significant. Joe, does that answer your question? Hopefully.
Yeah, I don't know if you could hear me, but
Oh,
it was a slide we were looking at previously. I think it was predictions that you were looking at, but it shows a similar pattern here.
Okay, great. Yeah, we
Thank
saw
you.
the same thing in the predictive model as well.
Okay.
Yeah.
Why don't we talk that to the other Wall Street, all that,
Mhm.
all those practices. In the past 30 years, have any of these treatments become less disruptive to the forest, like has bigger, better tires, labor? Like, would you think if you repeated those experiments now?
I doubt it, to be honest. I think that a lot of they use a lot of the same types of equipment, and generally forestry equipment, it trends towards generating more revenue and being more efficient, being able to, carry more stuff, for lack of a better term, rather than being less impactful to like wildlife.
Do you know for say that if they did their harvesting in the summer or the winter? Because it sounds like the may have been less damage if they did.
Dr. Haas?
But it was, they were mostly harvested.
Yeah.
Any individual dispersion that a plethodontid uses like that?
Yeah, so they don't really go very far. Generally, like most of the dispersal happens when they're very young. So like the mommy salamander will lay her clutch of eggs in, a terrestrial environment, and then the babies will all disperse out from their natal grounds and seek out a nice moist place for them to kind of inhabit. And usually during like drier periods, they will be more territorial. And claim territories by using scent marking. So then they don't really travel that far. They have really high site fidelity. So even if they are displaced from the site that they've kind of claimed, they will go back. So once they disperse from their natal grounds, they don't really go very far. And the dispersal distance, like I think the average was about the size of their home ranges, and the home range size is only about the size of a log. So they don't really go that far.
Is this it for SASAB? Do we know, is there any, what else, what would another decade do for us in terms of salamanders?
We need to go 100 years, right?
Yeah, we gotta rival MOFEP. MOFEP is the other long-term salamander study in Missouri, and their study is supposed to be 100 years. So hopefully the technicians will, all the undergrads will keep going for SASAB. Yay! Yeah.
We're still looking for silvicultural.
I have a question about SASAB too. This is just very general because I'm from the West, and when I think clear cut, I think like the entire mountainside, right? And what are the other impacts on wildlife? Are you guys can you speculate about that?
Yeah, so some animals love clear cuts, like the moose, the deer. I know in Michigan, they hate like the moose and the deer because they always come into the clear cuts and eat all the seedlings that they plant or the seedlings that are just naturally regenerating. They're like, ah, these darn moose. And then like reptiles love clear cuts because they like the heat, they like basking. So reptiles, if there's usually like a like silvicultural study on like herpetofauna broadly, the reptile is always really happy in the clear cuts. They're like, wow, reptile abundance increased. But amphibians generally don't like it. They like it cool and moist, and usually in most treatments, the temperature will increase in some way or the moisture will decrease in some way, so they don't like it. And then like small mammals, I think I'm forgetting the exact, I think it varies, but there are some species that are definitely drawn to clear cuts and they really like that habitat, and other species not so much. So it just depends on like who you're managing for, what goals. It's not necessarily that like all clear cuts are evil and bad. It's just, it's a management tool. And if you're managing for salamanders, probably don't do that. But anything else, like, sure.
You know, better than small neighborhoods.
Yeah, the plant studies are really cool from SASAB. Any other questions? Yeah.
In the study working on the same site. A little bit ironic. Silvicultural treatment that worked best to generate growth was the shelter. So again, depending on what you're trying to manage.
Yeah.
Zoom and close up, so Tori's defense for faculty, they're welcome to attend, it will be from it's starting at 1 p.m. in 136 feet, Cheetos, the big conference room. Cheeto, and I thank you all for attending.
Yeah.
Thanks.
Thanks for coming.